I'd like to talk to you about some of what we're doing in a much less biodiverse system in Florida. And Mote Marine Laboratory is an independent nonprofit dedicated to marine science, conservation, and education. We focus most of our work on Florida's coral reef. It's the third largest barrier reef in the world. It extends about 563 kilometers off of the southeast coast of the United States. It's very economically important, supporting more than 70,000 local jobs just in South Florida alone, and responsible for generating billions of US dollars a year in revenue. Just recently at a coral reef off of Key West, where even in its horrendously degraded state, it was still able to repackage and redirect about 97% of the wave energy that came in from offshore during Hurricane Ian. Our coral community in Florida's coral reef has gone from something like 40 to 60% live coral cover to now less than 2% coral cover. So it is a horribly degraded reef ecosystem, which makes my job very fun. We need to find a way to return ecological function and some semblance of form to that ecosystem. We've taken a lot of our cues from silviculture and forestry, and we even call these coral nurseries, and we even call these coral trees, where we propagate fast-growing weedy species, and this is one of the 2 or 3 or apparently 10 species of Acropora that we have in the Caribbean. In large numbers, and then vegetatively propagate fragments from these colonies and then outplant them onto the reef. However, Mote has also really strongly focused on the much more rich suite of species in the massive boulder brain and star coral species with a technique called microfragmentation. So one of the reasons that many groups do not focus on massive form corals is because they grow so slow. But microfragmentation, when you cut these into really tiny pieces, it basically initiates a much faster growth response, so 40 to 50 times faster growth. This colony was outplanted in that method in 2015, and this photo was taken in 2020. So in just 5 years, we saw microfragments outplanted onto the reef fuse and reproduce. We've also invested quite a bit in selective breeding. So taking those resilient individuals, so thermally tolerant, disease resistant, tolerant of ocean acidification conditions, and crossing those parents to produce offspring that have multiple traits. We have 92 permitted, roughly 3 square kilometer restoration sites, which amounts to about 250 square kilometers of restoration area. To service those locations, we operate 3 land-based coral nurseries, one here in Summerlin Key, where my office is, and then one here in Isla Morada, and one in Key Largo. And then each of those is paired with an offshore coral nursery, along with one down here off of Key West. But as we found very quickly over the years, that's not enough. So we're adding 2 more land-based coral nurseries to increase the supply of genetic material for restoration. We've still managed to outplant over 258,000 genetically resilient cultured coral colonies onto Florida's coral reef, and we're set to increase that number dramatically over the next really 4 or 5 months. Most of those corals that we've put out, we've approached much in the way that silviculture was taken, where you come in after a disturbance, you take a species and lay it out in relatively uniform distribution to create a forest where there once was one. But that's not a natural assemblage of species. So what we're trying to get to is if we can, literally a random assignment of the location of each of these species and genotypes within each of those species in the restoration plot. Florida's coral reef is almost depauperate of corals, but it is phenomenal growing environment for macroalgae and algal turf and long sediment-laden algal turf. It's an algal playground. So to facilitate any sort of recruitment on our reefs, we have to look at increasing grazing intensity. And we tested this in the field about a decade ago. And chose some of these very algal dominant patch reefs in our lagoon And through some out there as controls, some we added crabs at about 1 crab per square meter, and then some where we removed the algae and then put crabs on there to see if they could just keep it down. Just by adding crabs or by removing the algae and then adding crabs, we affected an 85% reduction in the cover of macroalgae on these reefs, which led to a 3 to 5-fold increase in the abundance and richness of reef-associated fishes, and more surprisingly and interestingly, a 2 to 4-fold increase in the recruitment. Every year, we're outplanting more and more species and more genetic individuals within each of those species to affect some fairly high density outplanting on each of these plots. And then on a subset of those, we're actively not outplanting, we're stocking cultured crabs onto those plots to affect an increase in the grazing intensity, to look at facilitation and some successional forces on these plots, as well as looking at the accretion potential through time. Keeping track of who's who on a reef where you're repetitively outplanting onto that same plot becomes a bit of a challenge. So we've used the imagery to build some of these digital maps of who's who. These are genetic individuals that we've outplanted through time. We are actively trying to integrate some machine learning, machine vision, and artificial intelligence into not only the imagery capture, segmentation, and classification pipelines, but then also into trying to develop synthetic data to train some of these algorithms, and eventually, with some partners at Duke University, build in some hydrodynamic modeling components, and then with partners at Stony Brook University, build in some connectivity modeling components that allow us to not only predict what we should and should not be putting onto the reef, but where should we be putting it so that we're affecting a source for a target sink, and then where should we be putting the crabs to make sure that that sink is appropriate for the settlement of the larvae that are coming in.